Guys, is my screen visible? Can someone unmute and let me know if I'm audible and if my screen is visible?
Yes, madam.
Okay, thank you so much. Okay, so uh, my screen is visible and I'm also audible, so it's live caption. Yes, so I'll start today's session. So, good afternoon, everyone. My name is Aishi and I'm a second year PhD student from the Department of Chemistry, IIT Bombay, and I'm your NPTEL TA for this course. And this is the last live session for my part of this sessions. You'll have one more live session with another TA, I think this Tuesday. But we needed to discuss some of the remaining questions, so I thought that we could get a summary session also. So in this session also, we'll be discussing some assignment questions from this year that you've already submitted, or from some sample questions from last year also, and hopefully that will help you in the exam. Solving questions will help you in the exam. So moving on to the first question. One second. Yeah. Which one of the following best describes genome assembly in bioinformatics? The options are predicting the functions of genes based on their sequence similarity to known genes, determining the order and orientation of DNA fragments to reconstruct the original sequence of a genome, aligning RNA sequences to a reference genome to determine differential gene expression, identifying and characterizing mutations in the genome sequence. Anyone?
Option 2.
Option 2, determining the order and orientation of DNA fragments to reconstruct the original sequence of a genome. Okay, anybody else? Okay, yes. So the correct option is option 2, determining the order and orientation of DNA fragments. So let's learn a little bit about genome assembly. So genome assembly is basically a process of reconstructing the complete genome sequence of an organism from the short DNA sequences generated by high throughput sequencing technologies. And the goal of genome assembly is to obtain a contiguous and accurate representation of the entire genome of an organism. Okay, so the process of genome assembly involves several steps, including pre-assembly processing, sequence assembly, and post-assembly processing. And in pre-assembly processing, the raw sequencing reads are subjected to quality control, trimming, and filtering to remove the low-quality reads, adapter sequences, and contaminating sequences. The resulting high-quality reads are then subjected to sequence assembly. And in sequence assembly, the reads are aligned to a reference genome or assembled de novo using various algorithms and software tools. De novo assembly involves the construction of a De Bruijn graph, or basically overlap graph, to identify the overlapping regions between the reads, which are then merged into contigs. And the contigs are then scaffolded using paired end or made pair reads to generate large sequence fragments or scaffolds. And the scaffolds are further polished using various error correction tools and consensus calling algorithms to improve the accuracy of the assembly. In post-assembly processing, the assembled genome is evaluated for completeness, accuracy, and consistency using various metrics and tools. And the assembly can be annotated with gene models and functional annotations to facilitate downstream analysis. Now, genome assembly is a complex and computationally intensive process that requires expertise in bioinformatics and computational biology. The quality of the assembly depends on various factors, okay, including the sequencing depth, read length, sequencing technology, assembly algorithm, and the quality of the input data. So if we go back to this question, does genome assembly actually predict the functions of genes based on their sequence similarity to known genes? So genome assembly does not directly predict the functions of genes based on their sequence similarity to known genes. Genome annotation is the process of identifying genes, regulatory elements, and other functional elements in the assembled genome. This annotation process usually involves the use of computational tools to predict gene structures and functions based on the sequence similarity to known genes, as well as experimental validation to confirm the accuracy of the annotations. However, even with the best genome assembly and annotation methods available, there can be limitations and uncertainties in predicting So even with best genome assembly and annotation methods available, there can be limitations and uncertainties in predicting gene functions solely based on the sequence similarity. So additional experimental studies are often needed to fully understand, the functions of genes in an organism. So this first option is not directly accurate. Okay. So the second option, that's the correct option. Does genome assembly determine the order and orientation of DNA fragments to reconstruct the original sequence of a genome. This is correct, because genome assembly is the process is of it's a process means piecing together DNA fragments obtained from sequencing into a contiguous and accurate representation of the original genome, and this process involves determining the order, orientation and overlaps between the fragments to reconstruct the original sequence. In other words, genome assembly aims to put the sequenced fragments back into the correct positions along the genome, like putting together a puzzle, and the resulting genome assembly can then be used for downstream analysis such as gene annotation, comparative genomics and functional studies. Okay. Next we come to option 3. Does genome assembly align RNA sequences to a reference genome to determine differential gene expression? No. Genome assembly is not typically used to align RNA sequences to a reference genome to determine differential gene expression. It's just that you piece together the DNA fragments obtained from sequencing into a contiguous and accurate representation of the original genome. So while what we call RNA seq. So while RNA sequencing can be used to study gene expression and transcriptome analysis, it typically involves aligning RNA sequence reads to a reference genome or transcriptome to identify gene expression levels and differential expression. So So while RNA sequencing can be used to study gene expression and transcriptome analysis, it typically involves aligning RNA sequence reads to a reference genome or transcriptome to identify gene expression levels and differential expression. So these analyses can then be used to study gene function, identify differential expressed genes between different conditions or treatments, and other downstream analysis. So this option is also not correct. The fourth option, does genome assembly identify and characterize mutations in a genome sequence? So assembly can be used to identify and characterize mutations in a genome sequence, but it is not always the most appropriate method for doing so. So once a genome is assembled, it can be compared to reference genome or other assembled genome to identify differences in sequence, such as single nucleotide polymorphisms or SNPs, insertion, deletions, or large structural variants. However, the accuracy of identifying mutations in a genome assembly can depend on a variety of factors, including the quality and the completeness of the reference genome, or the accuracy and the coverage of the sequencing data. Therefore, other tools such as variant calling pipelines or comparative genomics methods may be used in addition to genome assembly to identify and characterize mutations in a genome sequence. Okay, so all in all, the second option is the correct one. I've already explained. So all in all, in bioinformatics terms, genome assembly represents the process of putting a large number of short DNA sequences back together to recreate the original chromosomes from which the DNA originated. And sequence assembly is one of the basic steps after performing next generation sequencing or nanopore sequencing. So the established genome assembly can be submitted then to databases such as the European Nucleotide Archive or the NCBI assembly and ensemble genomes. And you can basically also browse these databases for genome sequences done by other researchers. Okay, so there are basically two types of genome assembly, different types of genome assembly: de novo assembly and mapping to a reference genome, also known as reference-based alignment. So de novo assembly refers to the genome assembly of a novel genome from scratch without the aid of reference genomic data. And a reference genome or reference assembly is a digital nucleic acid database acting as a representative example of a species set of genes. So once the reference genome is available, with its aid, the genome assembly becomes much easier, quicker and even more accurate. And therefore, unless necessary, researchers choose the method of reference-based alignment, because reference-based alignments has become the standard in diagnostics. And the advantages of reference-based alignment will be that they are good for single nucleotide variations and small indels. They work for deletions and duplications by using coverage information. It's a quick method to assemble the genome. It hides the raw data limitations. There are more tools to work with the results, and it's easier annotation and comparison. However, the disadvantage of reference-based alignment is that it requires a reference genome. Obviously, that's a big limitation. Also, it's limited by the read length for feature detection. Now, the advantages of de novo assembly will be that it does not really rely on a reference genome, so that's a limitation that's not there. And it's used to search unknown genes or transcripts, such as transcripts with new introns, chain splice sites, etc. And it's very good for structural variations. However, the disadvantages are that it requires very high quality, data, raw data, and it's a slow method and requires high infrastructure. Okay, so how do you evaluate an assembly? After the genome assembly, it's important to evaluate the quality of an assembly, and there can be some factors that will help you. Like there are some terms such as N50, which means half of the genome sequence is larger than or equal to the N50 contact size, or coverage, or average contact length. So these are some of the factors that you might want to read about if you're fascinated about it. And in addition to the process of genome assembly, there are also some other factors that can strongly affect the quality of genome assembly, like paired end sequencing and long read technology are two strategies to improve the quality of genome assembly. So the factors that might affect or hamper genome assembly is: number one, genome size. The bigger the genome is, the more data is needed. So therefore, before ordering sequence data, you need to estimate the genome size, which may be inferred by investigating the genome size of a very closely related species. Also, repeats, amount and distribution of repeated sequences in a genome largely influence the genome assembly results, and this can lead to misassemblies and an incorrect estimate of the size of the repeats. The heterozygosity. If a genome is highly heterozygous, sequence reads from homologous alleles can be too different to be put together, probably leading to a more fragmented assemblies or creating doubt about the homology of the contigs. Obviously, ploidy level. So, if possible, it's better to sequence haploid tissue, avoiding problems caused by heterozygosity. And GC content. So, inhomogeneous GC content can cause a problem for Illumina sequencing, resulting in very low coverage in these regions. Okay, so what happens generally? First, nucleic acid, the process, if I have to say in brief, at first nucleic acid extraction is done. So for the DNA isolation or RNA isolation, people should remember a couple of things, that you should handle these nucleic acids very carefully, because DNA RNA integrity, DNA RNA purification, sufficient DNA RNA amount, very important terms. And compared with resequencing, de novo sequencing requires very superior quality nucleic acid. Okay, the most important nucleic acid quality parameters for next generation sequencing are chemical purity and structural integrity. Also, there are sequencing methods. So determination of sequencing methods is an important factor that influences the cost and success of genome assembly. Okay, so next generation sequencing has been applied to as many in many remarkable projects such as the 1000 Genomes project and even the Human Microbiome project. However, some important genes of interest still cannot be assembled correctly, mainly due to the interruption of report elements. Third generation sequencing is a promising solution to this problem based on long reads that span the repetitive regions. And also another important factor is raw data processing. So although there are assembly tools that prefer dealing with the raw data, including potential adaptive sequences, researchers highly recommend that people study the manual to determine whether the program requires quality trimmed data or not. So if data trimming is required, it would be necessary to omit poor quality data by trimming low quality read ends and filtering of low quality reads. So there are multiple tools, I guess, that are available for this purpose, such as Trimomatric or Prince. So if you are interested in knowing how exactly genome assembly is performed, you can maybe try reading out some papers. There are a lot of review papers online. So the answer will be the second one, correct. She's correct. So moving on to the next question. This is the next question. Statement A. BLAST is used in bioinformatics and molecular biology research to identify homologous sequences and infer functional and evolutionary relationships between them. Statement B is SINA is optimized for aligning large data sets and provides high quality alignments suitable for downstream analysis, such as phylogenetic inference and taxonomic classification. Which of the following options do you think is true regarding the authenticity of the statements? Anyone? Which statement do you think is true? Which statement or statements do you think are not true? Okay, both are true. Anybody else? Okay, so yes, both are true statements. So let's talk a little bit about BLAST. So BLAST is a widely used bioinformatics tool. This is not BLAST, it's widely used bioinformatics tool basically. The full form of BLAST is Basic Local Alignment Search Tool. It's widely used in bioinformatics, that is, and it's basically used for search of similar sequences in databases. And it is primarily used for identifying homologous sequences and inferring functional and evolutionary relationships between them. Now, BLAST searches can be used to compare DNA, RNA, or protein sequences, and the results can provide insights into the function and structure of unknown sequences by identifying similar sequences that have already been characterized. Okay, so it's basically a very fundamental tool in bioinformatics and molecular biology research, and its broad applicability has made it an essential tool in many fields of biology, including genomics, proteomics, and evolutionary biology. Also, it's freely available. You can take any sequence from NCBI, FASTA, any sequence, and you can actually put in BLAST and you can try seeing what are the hits that you get. Now, SINA or SINA is basically optimized. So what is What is the question? It's optimized for aligning large data sets and provides high quality alignments suitable for downstream analysis such as phylogenetic inference and taxonomic classification. So this statement is true. SINA is basically Silva Incremental Aligner. It's a software tool that is commonly used in bioinformatics to align large data sets of ribosomal RNA, rRNA gene sequences. So the tool is optimized to provide high quality alignments that are suitable for downstream analysis such as phylogenetic inference and taxonomic classification. So SINA or SINA uses a combination of incremental and progressive SINA or SINA, however you call it, incremental and progressive alignment strategies to basically align large datasets efficiently. And the tool is specifically designed to handle the large and complex datasets of rRNA or ribosomal RNA gene sequences commonly used in microbial ecology and phylogenetics research. Okay, and the resulting alignments are high quality and very accurate, which makes them ideal for use in downstream analysis. Okay. So overall, SINA is a very powerful tool for bioinformatics researchers who need to align large data sets of rRNA gene sequences. Its ability to provide high quality alignments is suitable for phylogenetic inference and taxonomic classification, makes it a very valuable resource for microbial ecology specifically. Okay, and evolutionary biology also, and other fields that may rely on molecular sequence data. Okay, and it is optimized for accuracy and speed, and it uses a combination of, as I said, incremental and progressive alignment strategies to align sequences efficiently, and the resulting accurate result alignments can be used for downstream analysis such as phylogenetic inference and taxonomic classification. Okay, so in summary, BLAST is a versatile tool for comparing biological sequences, while SINA or SINA is a specialized tool for aligning large data sets of ribosomal RNA or rRNA sequences, and both tools are widely used in bioinformatics and can provide valuable insights into the function and evolution of molecular sequences. Okay. Moving to the next question, what is the main advantage of high throughput sequencing over traditional Sanger sequencing? Anybody? Okay. Anybody else? Yes, so it's the answer will be high accuracy and longer reads. So what is the main advantage that high throughput sequencing brings that we crave for it over traditional Sanger sequencing? So the main advantage over traditional Sanger sequencing is the ability to generate large amounts of sequence data rapidly and very cost effectively. So Sanger sequencing, which is also known as the dideoxy sequencing, was the very first method developed for sequencing DNA and is still quite widely used till today. However, Sanger sequencing has several limitations, including its relatively low throughput and very high cost per base. So Sanger sequencing typically generates reads for 700 to 1000 bases, which is not exactly sufficient for sequencing entire genomes to large gene families. In contrast, high throughput technologies such as Illumina and PacBio sequencing can generate millions to billions of reads per run, with read lengths ranging from a few hundred bases to tens of thousands of bases. And this allows researchers to actually sequence entire genomes or transcriptomes, as well as to perform metagenomic analysis and study complex genetic variation. And high throughput sequencing has also enabled the development of new applications in genomics such as single cell sequencing, epigenomics, and metagenomics. So these applications were not feasible with traditional Sanger sequencing before due to their low throughput and high cost. So overall, the main advantage of high throughput sequencing over traditional Sanger sequencing is definitely its ability to generate large amounts of sequence data rapidly and cost effectively. It has revolutionized many areas of genomics and molecular biology research. So is it high accuracy and longer reads? So while it is true that some high throughput sequencing platforms such as PacBio sequencing can generate longer reads than traditional Sanger sequencing, the main advantage of high throughput sequencing over Sanger sequencing is not necessarily longer leads, but rather higher throughput and lower cost per base. So this is true, but the most important advantage will be higher throughput and lower cost base per base. So in terms of accuracy, the error rates of high throughput sequencing platforms vary depending on the technology used and specific application. For example, Illumina sequencing typically has a lower error rate than PacBio sequencing, but both technologies have improved significantly in the last few years. And Sanger sequencing still has the highest accuracy of all sequencing methods, but its lower throughput and higher cost per base limits its lower throughput and higher cost per base limit its utility for large scale sequencing projects. So therefore, while high throughput sequencing platforms may not always generate longer reads or be more accurate than Sanger sequencing, Their ability to generate large amounts of sequencing data rapidly and cost effectively has made them a very powerful tool for many areas of genomics research, including whole genome sequencing, transcriptomics, and metagenomics. Okay, so in principle, the concepts behind Sanger sequencing and next generation sequencing technologies are similar. So in both next generation sequencing and Sanger sequencing, it is also known as the dideoxy or capillary electrophoresis sequencing. The DNA polymerase adds fluorescent nucleotides one by one onto a growing DNA template strand, and each incorporated nucleotide is identified by its fluorescent tag. The critical difference between Sanger sequencing and next generation sequencing is sequencing volume. So while the Sanger method only sequences a single DNA fragment at a time, next generation sequencing is massively parallel, sequencing millions of fragments of simultaneously per run, and this process translates into sequencing hundreds to thousands of genes at one time. So next generation sequencing also offers greater discovery power to detect novel or rare variants with deep sequencing. So the benefits of next generation sequencing are definitely higher sensitivity to detect low frequency variants, and obviously faster turnaround time for high sample volumes, the comprehensive genomic coverage, lower limit of detection, higher capacity with sample multiplexing, and the ability to sequence hundreds to thousands of genes or gene regions simultaneously. So obviously the benefits of Sanger sequencing will be it's a familiar workflow. It is fast, cost-effective sequencing, but for low number of targets. Okay, and next-gen sequencing will be higher sequencing depth, enables higher sensitivity down to even 1%, higher discovery power, higher mutation resolution, more data produced with the same amount of input DNA, higher sample throughout. So what are the limitations of or challenges of Sanger sequencing? Low sensitivity. The limit of detection is like 15 to 20%. Low discovery power, not as cost effective for high numbers of targets. Low scalability due to increasing sample input requirements. While the challenges or the limitations of next generation sequencing will be less cost effective for sequencing low numbers of targets, like 1 to 20 targets, and time consuming for sequencing low numbers of targets. Okay. So in addition to higher throughput and lower cost per base, high throughput sequencing or the HDS has several other advantages over traditional Sanger sequencing as well. So there's higher sensitivity, as I said, like high throughput sequencing basically can detect rare variants and low frequency mutations that may be missed by traditional Sanger sequencing due to its lower depth and coverage. Other advantage is its ability to detect epigenetic modifications. So some high throughput sequencing technologies such as bisulfite sequencing, can actually detect epigenetic modifications such as DNA methylation, which are important in gene regulation and disease. Also improve speed. High, high, high throughput sequencing platforms can generate results much faster than traditional Sanger sequencing, which can take several days or even weeks to complete, and obviously increase flexibility. So high throughput sequencing platforms offer greater flexibility in terms of the types of samples that can be processed and sequenced, and the applications that can be performed. For example, single cell sequencing and metagenomics analysis are not feasible with traditional Sanger sequencing due to their low throughput. So overall, high throughput sequencing has a lot of advantages. It has basically transformed genomics research by enabling large scale sequencing projects and new applications that were not feasible with traditional Sanger sequencing. Okay. So yes, the right answer is higher throughput, higher accuracy, and longer reads. Some of the advantages I've already talked about these, but these are also some of the advantages that you can check for Sanger and the next generation sequencing, and also the disadvantages are also over here. Moving on to the next question. This DASH is a publicly available database that stores DNA and RNA sequence data and associated metadata, such as sequence annotations and literature references. Which one do you think is the correct answer?
GenBank.
Anybody else? Yeah. So the correct answer is GenBank. Have you ever heard or worked with the other databases? Because I have not, and these were new for me also. I have heard GenBank is very popular, so most of us must have worked with GenBank, but I have not heard about these three. So it was fascinating to read about these three also. So the correct answer is GenBank, obviously. So GenBank is a database of genetic sequence data maintained by the National Center for Biotechnology Information, or the NCBI. Okay, it's a part of NIH, National Institute of Health, in the United States, and it's a publicly available resource that provides annotated DNA and RNA sequences for a wide variety of organisms, ranging from viruses and bacteria to plants and animals. So GenBank contains millions of genetic sequence records submitted by researchers from all around the globe. And each record includes a nucleotide sequence, information about the source organism, and annotations that describe the features of the sequence, such as genes and regulatory elements. So researchers can use GenBank to search for sequences that are similar to a particular sequence of interest or to download sequence data for further analysis. So the database is essential tool for many areas of biological research, including molecular genetics, evolutionary biology, and bioinformatics. So this is what GenBank is all about. If we talk about the other, I had forgot to add pictures anyway. If we talk about the other three terms. So what is MacVector? MacVector is basically a comprehensive bioinformatics software package designed for molecular biologists and geneticists. So it is used to analyze, manipulate, and visualize DNA and protein sequences. So MacVector was first released in 1986 for the Macintosh operating system and has since become one of the most popular bioinformatics tools available for the Mac platform. Okay, and MacVector provides a wide range of features for sequence analysis, including sequence alignment, restriction analysis, primer design, gene annotation, protein analysis, and plasmid construction. It also includes a suite of molecular cloning tools that allow users to design, annotate, and simulate the construction of recombinant DNA molecules. So MacVector offers a user-friendly graphical interface that makes it easy for researchers to work with complex genetic data. Okay, and it also has an extensive documentation library, online resources, and technical support to help users with the analysis. So MacVector is widely used in academic and industrial research labs for a variety of applications, including genetic engineering, gene therapy, and drug discovery. And it is constantly updated and maintained by its developers, ensuring that it remains a very powerful and reliable tool for molecular biologists, basically. So, what is Adobe Gene? I am not like generally we pronounce Adobe Photoshop, so I'm guessing it's something like Adobe Gene also only. So I have not really heard about this software tool. I don't know if it's existent also. I'll look into more into it. But let's move on to KIME, or I don't know how it's pronounced. I think it's KIME only. So KIME's full form will be Quantitative Insights into Microbial Ecology. It's an open source bioinformatics software package that is used for analyzing and interpreting microbial communities, particularly those found in environmental samples such as soils, water, and human microbiomes. And it was initially developed in 2010 by researchers at the University of Colorado at Boulder, and is now maintained and further developed by a community of developers. So KIME offers a suite of tools for processing and analyzing DNA sequencing data, including quality control, demultiplexing, sequence alignment, taxonomic classification, and diversity analysis. And it supports multiple sequencing platforms, including Illumina, Ion Torrent, and 454. And KIME basically employs a series of algorithms and statistical methods to analyze the composition and diversity of microbial communities. It can visualize and compare the taxonomic composition of samples, identify differentially abundant taxa, and perform diversity analysis such as alpha and beta diversity. So KIME also allows users to perform predictive functional analysis of microbial communities, providing insight into the metabolic capabilities of the community. So KIME is widely used in microbial ecology research, and its applications include studying the human gut microbiome, understanding the effects of environmental changes on microbial communities, and identifying biomarkers for the disease. And the software is free and open source, and its source code is available on GitHub. If you are interested, you can actually check it out, making it accessible to researchers around the world. So KIME is also quite fascinating. But the correct answer for this question will be GenBank. So moving on to the next question. DASH is the study of proteins and their interactions with other molecules. It uses various techniques, including mass spectrometry and protein microarrays, to identify and characterize proteins and their functions. Okay. What do you think is the correct answer? One second. Yeah. Anybody? Proteins. Yeah. So it's quite self-explanatory. So when we talk about protein samples and proteins, it's proteomics. Genomics basically refers to the entire genomic sequence present in the particular system. Transcriptome refers to the entire transcript level, all mRNA transcript level that is current at that time present in the system. Bioinformatics we have already talked about, and cheminformatics we'll talk in one of the further questions. So since it is and also there's another thing called metabolomics, so which is also which talks about basically the small molecules or the metabolites in the system. There's something called lipidomics, where we talk about the lipids, all the lipids, the entire lipid pool of the system. So it's this omic studies are very fascinating and a very hot topic in today's time. So the study of proteins and other interactions will be proteomics. So yes, it's the correct answer. This is a very schematic format of showing proteomic analysis. So this is how you take a sample, any sample, it can be a tissue sample, cell sample or blood sample, and then you basically extract the protein. There are very complex protocols for that. You need to check papers and then optimize the protocol. Then you take out the sample, you take out the proteins, extract the proteins, you digest it, and then you put it in a vial, and then you do mass spectrometry with the sample, and then you can basically see data, and then with based on the peaks you can do further analysis. Okay. Moving on to the next question. What is the purpose of bioinformatic analysis in metagenomics? To assemble short DNA sequences generated by sequencing into longer contiguous sequences, to compare the DNA sequences in the metagenome to known sequences in databases, to identify and annotate the genes present in the metagenome, to determine the functional potential of the microbial community represented by the metagenome, or all of the above. What do you think is the correct answer? Anybody? Okay, anybody else? So yes, you're right. The answer is all of the above. So what is the purpose of bioinformatic analysis in metagenomics, right? So bioinformatic analysis plays a very crucial role in any kind of metagenomics analysis. It is the study of genetic material recovered directly from environmental samples containing multiple organisms. So the primary purpose of bioinformatic analysis in metagenomics is to analyze the massive amounts of data generated from high throughput sequencing technologies and to gain insights into the microbial communities present in the environmental samples. And the specific goals of bioinformatic analysis in metagenomics include the identification of microbial taxa, functional analysis of genes and pathways, and the reconstruction of the genomes and individual microorganisms. So some common bioinformatics tools used in metagenomics include sequence alignment tools, clustering algorithms, functional annotation tools, and machine learning algorithms. So bioinformatic analysis in metagenomics can help us to better understand the composition and the function of microbial communities, their interactions with the environment, and their role in various biological processes, such as nutrient cycling, bioremediation, and disease. And it can also lead to the discovery of new microbial species and enzymes with potential applications in biotechnology, medicine, and other fields. So does it assemble the short DNA sequences generated by sequencing into longer contiguous sequences? Yes. So one of the key tasks of bioinformatic analysis in metagenomics is to assemble the shorter DNA sequences generated by sequencing into longer contiguous sequences, and called contigs basically, and this process is known as metagenome assembly. So metagenome assembly involves taking the short reads generated by high throughput sequencing and assembling them into longer sequences that represent the genomes of the individual microorganisms present in the sample. And this is a challenging task because metagenomic data can be highly complex and very diverse, containing millions or even billions of short reads derived from a vast array of microorganisms. You're taking environmental samples, no, so vast array of microorganisms. So several different approaches are used to assemble metagenomic data, including de novo assembly, reference-based assembly, and hybrid assembly. So de novo assembly involves assembling the reads without using a reference genome, while reference-based assembly uses a closely related genome as the reference to aid in the assembly process. Hybrid assembly combines both approaches, using a reference genome to guide the assembly of contigs and then filling in gaps using the de novo assembly methods. So once the contigs are assembled, they can be used for downstream analysis such as taxonomic classification, functional annotation, and comparative genomics. And the quality of the assembly depends on the depth of the sequencing coverage, the complexity of the metagenome, and the accuracy of the sequencing technology and the assembly of algorithms used. So this option is correct. So does it compare the DNA sequences in the metagenome to known sequences in the database? Yes. So bioinformatic analysis in metagenomics often involves comparing the DNA sequences in the metagenome to known sequences in the database, and this process is known as sequence annotation or functional annotation. So sequence annotation involves identifying the functions and characteristics of the DNA sequence in the metagenome by comparing them to those in databases of known sequences, such as those already present in GenBank, as we saw in the last question, or even UniProt. So this can help to identify the taxonomic identity of the microorganisms present in the sample, as well as the functional roles of the genes and pathways they encode. So there are several different types of sequence annotation methods that can be used in metagenomics, including homology-based annotation, profile-based annotation, and machine learning-based annotation. So homology-based annotation involves comparing the sequences to a reference database using tools such as BLAST or HMMER. Now, profile-based annotation involves building hidden Markov models HMMs based on a set of known protein sequences and using these models to search for similar sequences in the metagenome. And machine learning based annotation involves training machine learning models on annotated sequence data, basically to predict the functions of new sequences. So sequence in annotation in metagenomics can actually provide important insights into the ecological and the functional roles of microorganisms in complex environmental samples and can actually help to identify potential targets for biotechnological applications such as the discovery of novel enzymes or metabolic pathways. Okay, so this option is also true. So does it identify and annotate the genes present in the metagenome? Yes. So one of the primary tasks of bioinformatic analysis in metagenomics is to identify and annotate the genes present in the metagenome, and this process is known as gene prediction or gene finding. So gene prediction involves identifying the locations of protein coding genes in the metagenomic sequence data as well as predicting their functions based on sequence similarity to known genes in the databases. So this can be a really challenging task, particularly in complex metagenomic samples where the DNA sequences of different organisms can overlap and be very difficult to separate. There are several different approaches to gene prediction in metagenomics, including homology-based methods, ab initio methods, and hybrid methods. Now, homology-based methods use sequence similarity to known genes to predict the locations and functions of genes in the metagenome. Ab initio methods use statistical models to predict gene locations used based on patterns in the DNA sequence itself. And hybrid methods combine both homology-based and ab initio methods to improve the gene prediction accuracy. So once the genes are predicted, they can be annotated with information about their functions such as enzyme classifications, metabolic pathways, and functional domains. And this can be done using tools such as InterProScan, which searches a variety of databases for functional information about protein sequences. And gene prediction and annotation in metagenomics can provide important insights into the metabolic potential and functional roles of microorganisms in complex environmental samples. And this information actually can be used in to better understand the ecology and the evolution of microbial communities as well as to identify potential biotechnological applications of the metagenomic data. So this option is also true. And the last option that we come across is, does it determine the functional potential of the microbial community represented in the metagenome? Yes. One of the key tasks of bioinformatic analysis in metagenomics is to determine the functional potential of the microbial community represented in the metagenome. So this involves identifying the functional genes and pathways present in the metagenome and inferring the metabolic capabilities of the microbial community. So functional potential analysis in metagenomics involves several steps, including gene prediction, functional annotation, and pathway analysis, all that we have talked about. And gene prediction basically involves identifying the protein coding genes present in the metagenome, while functional annotation involves assisting functions to these genes based on similarity to known genes in the databases. So pathway analysis involves identifying the metabolic pathways that are likely to be present in the microbial community based on the functions of the genes present. So there are several different tools and databases available for functional potential analysis in metagenomics, including MGRAST, KEGG, and MetaCYC. I don't know how it's pronounced. So these tools allow for the identification of metabolic pathways, the reconstruction of metabolic models, and the prediction of the physiological characteristics of the microbial community. So functional potential analysis in metagenomics can provide important insights into the metabolic capabilities of microbial communities in complex environmental samples as well as their ecological and evolutionary roles. So this information can be used to better understand the biogeochemical cycling of elements in ecosystems as well as to identify potential biotechnological applications of the metagenomic data. Okay, so that is why all of the above options are correct. So yes, she's correct. Moving on to the next question. Dash is used for taxonomic classification of microorganisms present in samples such as soil, air, and water. Anybody? Yes. Did anybody else write anything? Yes, Amardeep and Neha, both of you are correct. It's 16S. 16S rRNA. So the 16S rRNA sequencing is a very powerful tool for taxonomic classifications of microorganisms in various environments, including soil, water, and air. And this 16S rRNA gene is a part of the ribosome, which is essential for the protein synthesis in all living organisms. So this 16S rRNA gene contains both conserved and variable regions, making it an ideal target for PCR amplification and sequencing. So the process of using 16S RNA sequencing for taxonomic classification typically involves these steps. Like firstly, there's sample collection. So the first step is to collect a representative sample from the environment of interest, such as soil, air, water, or air. Then you extract the DNA. So DNA extraction is the next step. Basically, to extract DNA from the sample, and this can be done using a variety of methods such as bead beating, enzymatic digestion, or commercial kits. Then we go for PCR amplification. So once the DNA has been extracted, the 16S rRNA gene is amplified using PCR with universal primers that target conserved regions of the gene. And this amplification step generates many copies of the 16S rRNA gene, which can then be sequenced. And the second last step is sequencing. The amplified 16S rRNA gene is then sequenced using next generation sequencing technologies, and the resulting sequences are then analyzed using bioinformatics tools to determine the taxonomic identity of the microorganisms in the sample. And then the last step will be taxonomic classification. The final step is to assign taxonomic identities to the sequences based on their similarity to known sequences in databases such as NCBI's GenBank. And this can be done using a variety of software tools such as KIND, as we discussed in one of the questions, or even Megan. So overall, 16S rRNA sequencing provides a very rapid and accurate method for identifying the taxonomic composition of microbial communities in environmental samples, and can be used to explore the diversity of microbial communities in different environments, to track changes in community structure over time, and to compare the microbial communities in the different samples. So yes, this 16S rRNA answer is correct. Moving on to the next question. The interdisciplinary field that develops tools, algorithms, software to understand the biology by applying techniques from mathematics, computer science is known as statistics, and statistics is known as, anybody?
Bioinformatics.
Yeah, correct. It's known as bioinformatics, obviously. Sorry. Yeah. So bioinformatics is the field that combines computer science, statistics, mathematics, and biology, basically to analyze and interpret biological data. And the term bioinformatics refers to the development and application of computational tools and techniques for processing, analyzing, and interpreting large complex biological data sets, such as DNA sequencing data, gene expression data, protein structures, and metabolic pathways. So bioinformatics plays a very critical role in modern biology and biomedical research, and some examples can include genome sequencing and annotation. So bioinformatics tools are used to assemble and annotate genomes, identify genes and regulatory elements, and predict the functions of proteins. Then we have comparative genomics. So bioinformatics tools are used to compare the genomes of different species and identify evolutionary relationships, functional similarities and differences, and even potential drug targets. Then we have transcriptomics. So bioinformatics tools are used to analyze gene expression data and identify differentially expressed genes, functional pathways, and regulatory networks. Then we talk about proteomics. So bioinformatics tools are used to analyze protein structures and functions, predict protein-protein interactions, protein drug interactions, protein enzyme interactions, and identify protein RNA interactions, and identify their basically potential drug targets. And then we have metabolomics. So bioinformatics tools are used to analyze metabolic pathways and identify biomarkers for disease diagnosis, progression, and treatment. So in summary, bioinformatics is an interdisciplinary field that applies computational and statistical methods to analyze biological data and gain insights into the complex biological process. The field is very rapidly evolving, driven by advances in the high throughput sequencing technologies and the increasing availability of large and complex biological data sets. In the last few years, bioinformatics has really, last decade or so, bioinformatics has actually become one of the hot topics of research. So right, the answer is bioinformatics. Moving on to the next question, match the following. So you see some options in column A and some options in column B. Match them and let me know which one will be the correct answer. Anyone?
First option.
Anybody else? Yes, the correct answer is option is the first option. So what is metabolomics? Metabolomics is the study of small molecules or metabolites present in cells, tissues, and biological fluids. It is a rapidly evolving field that combines analytical chemistry, biochemistry, and bioinformatics to identify and quantify the metabolites present in biological systems and to understand their roles in cellular metabolism and disease. So metabolites are the end products of cellular metabolism and are involved in numerous cellular processes such as energy production, cell signaling, and biosynthesis. So metabolomics aims to measure and analyze the global metabolite profile of a biological sample to gain insights into the underlying metabolic pathways and their regulation. Okay, so the metabolomics workflow involves several steps such as sample preparation. So the first step is to prepare the biological sample for analysis. This may involve extracting and purifying the metabolites using various techniques such as liquid extraction, solid phase extraction, or protein precipitation. Then we go for metabolite profiling. So the next step is to measure and quantify the metabolites present in the sample using analytical techniques such as mass spectrometry, nuclear resonance, nuclear magnetic resonance spectroscopy, or gas chromatography. So these techniques can detect hundreds or thousands of metabolites simultaneously. Then there's data analysis. So the metabolomics data generated from these techniques and bioinformatics tools for analysis are complex and very require specialized bioinformatics tools for analysis, and the statistical methods are basically used to identify differentially expressed metabolites, metabolic pathways, and biomarkers that are associated with particularly biological process or diseases. And then comes interpretation. So the final step is to interpret the metabolomics data in the context of the biological system being studied, and this involves integrating metabolomics data with other omics data such as genomics, transcriptomics, and even proteomics to gain a comprehensive understanding of the cellular processes and regulation. So applications of metabolomics include disease diagnosis, drug discovery, and personalized medicine. For example, metabolomics can be used to identify metabolic biomarkers for early detection of diseases such as cancer, diabetes, and cardiovascular diseases. It can also be used to identify new drug targets and to optimize drug efficacy and safety. Moving on to genomics. So genomics is a study of the structure, function, and evolution of genomes, the complete set of genetic information of an organism. And genomes are made up of DNA, which contains the instructions for the development, growth, and reproduction of living organisms. And genomics involves the analysis of DNA sequence data to understand the genetic makeup and variation of different organisms, including humans, animals, plants, and microorganisms. So the field of genomics has grown rapidly in recent years due to advances in DNA sequencing technologies, which have made it possible to sequence entire genomes quickly and cost effectively. And the genomics workflow involves several steps such as DNA sequencing. The first step is to obtain DNA samples from the organism of interest and sequence the DNA using high throughput sequencing technologies such as Illumina, PacBio, or Oxford's Nanopore. Then we have genome assembly. So the next step is to assemble the raw sequence data into a contiguous genome using specialized software tools, and this involves aligning and overlapping the DNA fragments to reconstruct the entire genome. Then we have annotation. So the assembled genome is then annotated to identify genes, regulatory elements, and other functional elements. And this involves predicting the location and function of protein coding genes and non-coding RNA genes, and characterizing regulatory regions such as promoters and enhancers. Then we have comparative genomics. So the assembled and annotated genome can then be compared to other genomes to identify evolutionary relationships, functional similarities, and differences, and potential drug targets. Then we move on to data analysis. So the genomics data generated from these techniques are complex and require specialized bioinformatics tools for analysis. Statistical methods are used to identify genetic variations, disease-causing mutations, and genetic risk factors associated with particular diseases. So the applications of genomics include disease diagnosis, drug discovery, and personalized medicine. For example, genomics can be used to identify genetic mutations that cause diseases such as cancer, cystic fibrosis, and Alzheimer's disease. And it can also be used to identify new drug targets and optimize drug discovery, drug efficacy, and safety based on an individual's genetic makeup. Okay. Next, we move on to metaproteomics. So, metaproteomics is a subfield of proteomics that involves the study of all proteins present in the company's biological samples, such as microbial communities in soil, water, or the human gut, and it aims to identify and quantify the proteins expressed by the entire microbial community. Very similar to metagenomics in terms of proteomics aspect, and basically to understand the functional roles of these proteins in microbial metabolism, ecology, and interactions with their host organisms. So metaproteomics is also a very powerful tool for characterizing the functional diversity of microbial communities and their interactions with the environment. So the metaproteomics workflow includes several steps such as sample collection and preparation. So the first step is to collect the biological samples such as soil, water, or fecal matter, and whatever, and prepare it for analysis. And this may involve filtering, centrifugation, or other methods to concentrate the microbial cells. Then there is protein extraction and separation. The next step is to extract proteins from microbial cells and separate them by molecular weight or isoelectric point using techniques such as gel electrophoresis, or liquid chromatography. Then is protein identification. The separated proteins are then digested into smaller peptides and analyzed by mass spectrometry to identify the individual proteins. And this involves comparing the mass to charge ratio of the peptides to a protein database and using specialized software tools to match the peptides to their corresponding proteins. Then we have data analysis. The metaproteomics data generated from these techniques are complex and require specialized bioinformatics tools for analysis. And statistical methods are used to identify differentially expressed proteins, metabolic pathways, and functional modules that are associated with particular environmental conditions or microbial interactions. And then we move on to the last step, which is interpretation. The final step is to interpret the metaproteomics data in the context of the microbial community and its environment, basically. And this involves integrating metaproteomics data with other omics data such as metagenomics and metabolomics to gain a comprehensive understanding of the microbial community and its functional roles in the ecosystem. So applications of metaproteomics include environmental monitoring, bioremediation, and drug discovery. For example, metaproteomics can be used to identify the functional genes and pathways of microbial communities involved in soil remediation or wastewater treatment, and it can also be used to identify new drug targets and to optimize drug efficacy and safety based on the metabolic pathways of the microbial communities associated with particular diseases. Okay, so moving on to the last option, it is the microarray. So a microarray is a very high throughput technology used in genomics research to simultaneously analyze the expression of thousands of genes in a biological sample. It's a collection of DNA or RNA sequences called probes that are immobilized on solid surface such as glass slide or a chip. And the microarray workflow involves several steps such as probe design and synthesis. So the first step is to design and synthesize the probes. The probes are typically 20 to 70 nucleotide sequences that are complementary to the target genes of interest. Then we go on to sample preparation and labeling. The biological samples such as RNA or cDNA is extracted and labeled with a fluorescent dye or other reporter molecule to enable detection of gene expression levels. Then we go to hybridization. The labeled sample is hybridized to the microarray, which contains thousands of immobilized probes, and the sample binds to the complementary probes, and the fluorescence intensity of each spot on the array corresponds to the expression level of the corresponding gene. Then we move ahead to scanning and data analysis. The microarray is scanned to generate an image, and the fluorescence intensities are quantified using specialized software, and the gene expression data are then analyzed statistically to identify differentially expressed genes and to characterize the functional pathways and biological processes associated with the gene expression changes. So microarrays have been used in a wide range of applications, including gene expression profiling, genetic variation analysis, and diagnostic testing, and they are particularly very useful in studying complex biological systems such as cancer, where thousands of genes are involved in disease progression. So microarrays have also been used to study the molecular mechanisms underlying drug response and toxicity, and to identify biomarkers for disease diagnosis and prognosis. However, in the recent years, microarrays have been largely replaced by more advanced sequencing technologies such as RNA sequencing, which offer higher sensitivity and accuracy for gene expression analysis. Okay, so if you can ask me what is the difference between metaproteomics, like proteomics and metaproteomics. So proteomics and metaproteomics are two related fields of study that focus on the analysis of proteins only, but they differ in the scale and the complexity of the samples being studied. So proteomics is the study of the complete set of proteins expressed by an individual organism, tissue or cell type. It involves the isolation, separation, and then identification of the individual proteins as well as the characterization of their functions, interactions, and post-translational modifications. So proteomics techniques can provide insights into the underlying molecular mechanisms of biological processes, disease pathways, and drug targets. Metaproteomics, on the other hand, is the study of the complete set of proteins expressed by an entire microbial community in a complex environmental sample such as soil, water, or the human gut, and it involves the identification and quantification of proteins from multiple microbial species and the characterization of their functional roles and interactions within the community. And metaproteomics is a powerful tool for characterizing the functional diversity of microbial communities and their interactions with the environment. So in summary, proteomics focuses on the analysis of proteins in an individual organism or tissue, while metaproteomics focuses on the analysis of proteins in a complex microbial community within an environmental sample. Okay, so that is what metaproteomics is all about. Okay, so move with this. So the answer is one. First option. That is this one. You're correct. Moving on to the next question. Consider the following steps in metagenomics. Which one do you think is the correct answer? Anyone?
Option 4.
Option 4. Okay. Anybody else? So you're right. Option 4 is the correct answer. DNA extraction, shotgun, library preparation, genetic sequencing, alignment, and annotation. So how does this entire metagenomics process occur? So the first step is sample collection and DNA extraction. The first step in metagenomics is to collect the environmental samples such as soil, water, or feces that contains a complex mixture of microbial species. The DNA is then extracted from the sample using specialized protocols. Okay, you have to read papers and optimize the protocols. Then we go on to library for preparation. The extracted DNA is then fragmented and subjected to library preparation, where the DNA fragments are ligated to adapters that enable sequencing, and the resulting library contains a mixture of DNA fragments from multiple microbial species. Then we move on to sequencing. The library is then subjected to high throughput sequencing using next generation sequencing platforms such as Illumina or PacBio, and the resulting sequence data can generate millions to billions of short reads or fewer but much longer reads. Okay, and then we move on to quality control and pre-processing. Okay, the raw sequence data is then subjected to quality control and pre-processing steps to remove low quality reads, adapter sequences, and other artifacts that could affect downstream analysis. Then we move to taxonomic and functional annotation. The clean sequences, excuse me, the clean sequences are then annotated using specialized software tools that compare the sequences to databases of known microbial genomes or protein sequences, and this allows the identification of microbial taxa and their functional potential based on the presence of specific genes or metabolic pathways. And the last step will be data analysis and interpretation. So the annotated data can then further be analyzed using various statistical and computational tools to identify patterns of microbial diversity, community structure, and functional potential, and the results can then be visualized using various software tools and can be used to generate hypotheses about the roles of microbial communities in the environment. So these are the basic steps of metagenomics analysis. Yes. So moving on to the last question for today. So it's an algorithm to compare the primary biological sequence information such as the amino acid sequences of proteins or the nucleotides of DNA or RNA sequences. That's ribosomal RNA sequences. What is it? Which one do you think is the correct answer? Which one do you think is the correct answer?
Productivity.
Uh, which one do you think will be the correct answer for this question? It's an algorithm to compare the primary biological sequence information such as the amino acid sequences of proteins or nucleotides of DNA or RNA sequences. So what is it? Anyone?
Blast.
Anybody
Blast.
else? Okay, anybody else? Correct. So I already talked about BLAST in the last one of the last questions. I talked about what BLAST basically means. It's a basic local alignment search tool. Let's talk about FASTA. So what is FASTA? FASTA algorithm is basically a bioinformatics tool used for sequence alignment and similarity searches. It was first introduced by David J. Lipman and William R. Pearson in 1985 and has since become a very widely used tool in molecular biology and bioinformatics. The FASTA algorithm works by comparing two sequences, a query sequence and a target sequence, to identify regions of similarity. And the algorithm first identifies potential matching regions by identifying k-mer matches between the two sequences, where a k-mer is a short sequence of length k, and the k-mer size is typically chosen based on the expected level of similarity between the sequences. So once the potential matching regions are identified, the algorithm then calculates a score for each potential alignment based on the degree of similarity and the lengths of the aligned regions, and the algorithm then outputs the highest scoring alignments as potential matches. So one advantage of the FASTA algorithm is that it is relatively computationally efficient compared to other sequence alignment algorithms such as BLAST, but this makes it well suited for searching large sequence databases for similar sequences. So the FASTA algorithm has been used for a wide range of applications, including identifying new protein families, detecting homologous sequences across different species, and identifying conserved regions within a single protein family. What is the Velvet algorithm? So the Velvet algorithm is a de novo assembly algorithm used in metagenomics and genomics research. It is designed to assemble short reads generated by next generation sequencing platforms into contiguous sequences called contigs. And the Velvet algorithm works by first constructing a deep-brain graph from the input reads, and this graph represents all the possible short overlapping sequences of length k, which are called k-mers, found in the reads, and the edges between these k-mers represent the overlaps between the adjacent reads, and the k-mer size is typically chosen based on the read length and the complexity of the input data. So the Velvet algorithm then uses a series of heuristics to basically simplify the deep brain graph and to identify potential errors and ambiguities in the graph. And this simplification step involves merging similar nodes and resolving conflicts between alternative paths in the graph. And the algorithm then uses a simplified graph to construct contexts by traversing the graph paths and stitching together the k-mers. The Velvet allows for multiple rounds of iteration with progressively smaller k-mer sizes, which can help to resolve errors and ambiguities and to improve the quality of the final assembly. So the Velvet algorithm is widely used in metagenomics studies to assemble short reads from complex microbial communities, where it can help to identify novel microbial taxa and functional genes. It is also used in genome sequencing projects to assemble reads into complete or nearly complete genomes, particularly for organisms with small or repetitive genomes. So the answer will be BLAST. We have also talked about Mac vector. I'll talk once about BLAST again. So the correct answer will be BLAST. The basic local assignment search tool is basically a bioinformatics algorithm used to compare nucleotide or protein sequences against a database of known sequences. Was first introduced by Stephen Olshul and colleagues in 1990 and has since become one of the most widely used bioinformatics tools. So the algorithm works by breaking down a query sequence into short words of fixed length, which are called k-mers, and then searching a database for matches to those words. And this initial step also helps to identify regions of similarity between the query sequence and the database. So once potential matching regions are identified, the algorithm extends the search to identify longer regions of similarity by aligning the query sequence with the database sequence. And the algorithm uses a scoring system to assign a numerical value to each possible alignment, taking into account the degree of similarity between the two sequences and any gaps or mismatches in the alignment. The BLAST algorithm can then be run in several different modes, including a basic nucleotide or protein search, a pairwise alignment, and a multiple sequence alignment. And it also includes various advanced options such as filtering the database to exclude certain sequences or adjusting the scoring parameters to customize the search results. So one of the main advantages of BLAST is its speed and flexibility. It can quickly search large databases for matches to a query sequence and can then be used for a wide range of applications, including identifying homologous sequences, annotating genomes, and identifying gene function. So overall, the BLAST algorithm has revolutionized the field of bioinformatics and has become an essential tool for researchers in genomics and proteomics and other areas of molecular biology. So BLAST is the correct answer. To explain Mac vector again, it's basically a bioinformatics software package designed for molecular biology and sequence analysis. It was developed and distributed for Mac vector INC basically, and is available for use on all Macintosh computers. So Mac vector includes a variety of tools for DNA and protein sequence analysis, including sequence alignment, cloning simulation, plasmid map drawing, and primer design. It also includes a number of tools for advanced sequence analysis, such as restriction enzyme analysis, protein structure prediction, and phylogenetic analysis. So MacVector provides a user-friendly interface with graphical representation of sequence data and analysis results, and it also includes a variety of database search tools for searching public sequence databases such as GenBank, SwissProt, and BDB. Protein data bank. So overall, MacVector is a very versatile and powerful software package for molecular biologists and bioinformatics with a wide range of features and tools for sequence analysis and data management. Okay, but the correct answer for this question will be BLAST, as I explained. So this was the last question for today, and this was the last summary session. If you have any questions from the session that we did today, you can ask. So anyway, thank you. We'll conclude the session today. Thank you, everyone.